Alright, welcome back. In this video, we're going to go over how to alter the contractile force of the heart. In other words, what we're going to be talking about is how to make the heart contract more forcefully, with more strength. That does not necessarily mean speed. This is more like force. So first, let's go over the innervation of the heart. How does our brain, how does our body actually talk to the heart to tell it to contract more forcefully? Well, that requires the parasympathetic system and the sympathetic system. So heart physiology revolves around the parasympathetic system and sympathetic system. So let's talk about the parasympathetic system first. The parasympathetic system involves the vagus nerve. The vagus nerve is one of the cranial nerves. More specifically, it's cranial nerve number 10. The vagus nerve secretes acetylcholine, which is a neurotransmitter. Acetylcholine binds to muscarinic receptors located on the atria. So located in the left atrium and the right atrium. Now notice, muscarinic receptors are actually not located in the ventricles. They are absent in the ventricles. They're only located in the atria. The sympathetic system involves the thoracic spinal nerves. They release the neurotransmitter Norepinephrine. And this binds to beta receptors of the heart. More specifically, the beta receptors on the atria and the ventricles. At the same time, your adrenal glands produce epinephrine. So the adrenal glands make epinephrine and then they put it in your bloodstream. And the epinephrine travels in your blood until it reaches the heart. And then it binds to the beta receptors on the atria and the ventricles. So the first thing we should note is that there's muscarinic receptors only in the atria, but for beta receptors, they're in the atria and the ventricles. So all four chambers, the left atria, the right atrium, the left ventricles, and the right ventricles. So let's talk about actually how to alter the contraction of the heart and how forcefully. There's a lot going on, so let's take it slowly, really slowly. And it does look complicated, but you probably already know half of this. Because the first half is literally the GS pathway. I'll link a video in the description, and you can look at that GS pathway video. So let's start. The very first thing, so actually what we're talking about here is the muscle cell of the heart. So this is not the pacemaker. So this is not the SA node. This is not the AV node. This is not the Purkinje fibers. It's the actual muscle cells of the heart. So the first thing is norepinephrine or epinephrine binds to the beta receptors, the Beta-1 receptors on the muscle cell. This activates the G protein. Then it activates adenyl cyclase. Adenyl cyclase is like a factory. It turns ATP into CAMP. Then CAMP activates protein kinase A. So now protein kinase A, so here going forward is where things change. And this is actually where we're actually going to alter the contractile force of the heart. So protein kinase A does a lot of stuff. The first thing it does is it goes to the L-type calcium channels on the membrane and it opens them up, it activates them. So calcium can go inside the sarcoplasm freely. So it basically opens the L-type calcium channels. At the same time, protein kinase A is going to go over the sarcoplasmic reticulum. If you remember, this is like the endoplasmic reticulum, but it's called sarcoplasmic reticulum because we're in the muscle cell. And the sarcoplasmic reticulum is a house for calcium. So what the protein kinase A is going to do, it's going to go over the sarcoplasm and it tells the sarcoplasm, hey, it's time to release calcium. So it releases calcium into the sarcoplasm. Protein kinase A also goes and ramps up ATPase activity. So in this muscle contraction cycle video, I mentioned the contractile cycle of the muscle revolves around calcium and ATP, and ADP plus P. So in order for the myosin heads to actually bind to actin and cause a contraction, it requires ADP plus P. So when ADP plus P is bound to the myosin head, it's charged, and it's able to actually bind to actin and cause a contraction. After the contraction is finished, ATP comes along and binds to the myosin heads, freeing it from actin. And then it goes back into the same original position, and the ATP turns into ADP plus P with the help of ATPase. So what we're doing is we're ramping up this ATPase so we can make more ATP turn into ADP plus P, so they're charged and ready to bind to actin. The last thing that protein kinase A does is it tells something called the Surca pumps to let calcium in to the sarcoplasmic reticulum. So on the sarcoplasmic reticulum, there's something called the Surca pumps. And what this does, it shoves calcium back inside the sarcoplasmic reticulum. So protein kinase A is going to ramp this up. So you're probably thinking, wait a second, this doesn't make any sense. We let calcium go out of the sarcoplasmic reticulum. Why are we bringing it back in? Aren't we just undoing what we just did? Well, let's actually think about this for a second. Actually, we're going to come back to that. We're going to go here for a second. And we're just going to, let's do a little bit of an overview. So activating Beta-1 adrenergic receptors increases L-type calcium activity, increases calcium release from the sarcoplasmic reticulum, increases ATPase activity, so we can get more contractions and more forceful contractions, and we're going to do uptake more calcium by the Surca pump, and it goes back into the sarcoplasmic reticulum. And all of this does, it changes the force of the contraction, called inotropy. So we're increasing the contraction force. So and here's just a little recap. So now let's go back to this original thing we were talking about. Is that why are we doing this? Why are we releasing calcium and then shoving it back in? That serves no purpose, you're probably thinking. Well, it's actually very smart while we're doing this. So let's think about this. Let's think about a scenario. Imagine if you blocked the Surca pumps, so calcium cannot go back in the sarcoplasmic reticulum. So what's going to happen is that calcium is going to stay in the sarcoplasm. And remember what we talked about muscle contraction cycle in that video? I mentioned that calcium binds to troponin, which pushes tropomyosin away, which exposes actin binding sites, the myosin binding sites on actin. Well, if calcium is always present, those binding sites will always be exposed, and myosin will never want to let go of actin. It will permanently stay contracted. That's a really bad thing. Because remember, in the atria and ventricles, their goal is to fill up with blood and then contract so we can get the blood throughout the body. Well, if they're always contracted, we could never fill up those chambers with blood, and it would be a really bad thing. not enough blood would get to our brain. It would not enough blood will get to our organs. They will die because with lack of oxygen. So what we did is our body countered this by saying, all right, at the same time, we're going to shove some calcium back into the sarcoplasmic reticulum so we give our muscle cells a little break so it can relax, so we can actually fill the muscle cells, that atria and ventricles, with blood. And then we're just going to have it, calcium get secreted again. And it would go back into troponin, move tropomyosin away for another contraction. But we need a break. So that's why we need to sequester some calcium back into the sarcoplasmic reticulum so contraction is not always present. We don't want that muscle cell to always be contracting, permanently contracting. Think about when you go to a gym, for example, and you do bicep curls. Imagine taking that dumbbell, that 25-pound dumbbell, or whatever really weight you can maximum carry, and you just curl it for like 45 minutes straight. That would not be a good thing at all. Right? You won't be able to do that, most likely. But it would be a really bad thing. You need to rest. You need a break. Your muscles need a break. So same thing with your heart muscles. It needs a break so it can fill up with blood. So your atria and ventricles can fill up with blood. That's why it's super important. So this is how we actually can change the force of contractions. Make it more strong. More forcefully. So, if you found this video helpful, please like and subscribe. Until next time, later.